So I work in between the Oxford Eye Hospital and then the neuroimaging department. Overall, my research interests are how to recover sight at the eye. And I'm interested in finding out how a loss of input at the eye affects the visual cortex. If the brain doesn't get input, we expect the neurons in the brain to change. I'm looking at structural measures, neurochemical measures, and functional measures. And this is so important because currently there are treatments to try and restore the cells at the eye. And we want to know if the brain is actually ready to process that restored input, or if there is a barrier because the brain has changed. Let me know if they're comfortable. In the eye hospital, I learned how to use all the different vision measures. And also we have research that's investigating gene therapy for eye disease. We can collect all the vision measures in the eye hospital in the morning and then do MRI scanning in the afternoon. The neuroimaging facilities at NDCN are really cutting edge. We have 2 different MRI scanners, a 3 and a 7 Tesla one. We have new analysis techniques and incredible ways to image the human brain. We can work with physicists and design the methods that are best to collect our data. So I use a cutting-edge technique, which is only really available here, and we can measure neurochemicals at multiple voxels in the brain. Having that overlap between the 2 divisions means we can ask really interesting questions about how the eye and the brain work together. I recently gave a talk on my PhD work in a conference, and that was a career-changing opportunity for me. At the end, there was actually a queue of researchers waiting to ask me questions. I feel like having this experience from NDCN has made me a lot more employable for postdoc positions.